Chapter 3. Energy Information. Organisms cannot achieve the strategy of energy intake and lock-in without being connected to the environment. Individuals receive information from environmental substances or objects, and also send information proactively to the environment and receive it back through reflection. These messages are manifestations of energy, with each message representing a distinct form of energy. The energy carried by information is much weaker and more discrete compared to the energy that can be absorbed. It is referred to as microenergetic information in this book for the sake of simplicity. There is a difference between ingestible energy and microenergetic information. Ingestible energy refers to the energy selected and consumed by the organism from specific energy loci and sources, whereas microenergetic information refers to something else. Organisms receive microenergetic information from emitted energy of all substances or objects in their environment, which is passively received by them, with the exception of organisms that send out detection signals. For example, bats receiving ultrasonic waves proactively emit and reflect. Microenergetic information is included in electromagnetic waves, like microwaves, visible light, X-rays, gamma rays, etc., particle radiation, such as alpha rays, beta rays, neutron radiation, neutrinos, etc., acoustic radiation, for instance, ultrasound, sound waves, seismic waves, etc., natural small molecules, ions, compounds, and various biological homemade pheromones. Organisms evolve unique receptors to acquire microenergy information and establish appropriate information response mechanisms, which enhance their survival during energy intake and lock-in. Sensory receptors. One common functional characteristic of receptors in organisms is that each type is highly sensitive to particular stimuli. In other words, receptors require minimal intensity, sensory threshold, of a specific stimulus to elicit the corresponding sensation. This method of stimulation is known as the appropriate stimulus for that particular receptor type. For instance, retinal photoreceptor cells are most receptive to electromagnetic waves of specific wavelengths, while cochlear hair cells are adaptive to mechanical vibrations of specific frequencies. This implies that any alterations that happen in an organism's internal or external environment are always first detected by the corresponding receptor type. This phenomenon exists because organisms have developed receptors and supporting components with distinctive structures and functions over a long period of evolutionary time. This enables them to receive and measure changes in the internal and external environments, which affect their life activities. A vast range of receptors has evolved in organisms, including visual, taste, olfactory, and tactile receptors, as well as more sophisticated receptors that can detect temperature, electromagnetic fields, pressure, speed, and direction. Organisms instinctively collect information from their surroundings that aligns with their energy intake strategies using their unique receptors. The type of food, state, direction, and location of movement determine what appropriate predatory action organisms would take. For instance, a shark's nose is its most sensitive receptor, allowing it to detect minute amounts of blood emanating from miles away and track the scent to its source. Sharks possess an acute sense of smell, particularly towards the scent of blood in seawater, and their olfactory system can identify a concentration as low as 1 part per million, ppm, exceeding a dog's capability to smell. A minor bleeding from a wounded fish can attract it from afar. A shark's nose can detect the time difference between the arrival of odor molecules and their concentration through both nostrils, which helps determine the direction and distance of the food source. Organisms, particularly animals, have receptors that capture information which could interrupt their energy balance and trigger alertness, defensive reactions, evasive actions, or flight responses. For instance, when our hand comes into contact with a scorching item, the temperature receptors send messages to both the muscles and the brain to elicit the instinctive reaction of retreating. Similar to the biodiversity of the Earth, the locations, structures, and functions of receptors are diverse and varied. Some receptors receive visceral stimuli, such as those located in the skin that sense pain, temperature, and touch. Other receptors receive electromagnetic and acoustic stimuli, for example, electroreceptors, visual receptors, and auditory receptors. Further, some receptors receive chemical stimuli, such as olfactory and gustatory receptors. Sharks additionally possess the same electroreceptor organ as rays and stingrays, referred to as the ampullae of Lorenzini. The reticular system located at the tip of the shark's head detects slight changes in electric and magnetic fields, as well as temperature gradients. In addition, there are specialized receptors in the walls of internal organs and blood vessels that receive information from within the body. The evolution of these receptors contributes to the maintenance of various energy balances and crucial body functions. For instance, the vessel wall contains the carotid body and carotid sinus. The carotid body functions as a chemoreceptor, sensing changes in the oxygen and carbon dioxide levels, as well as the pH of arterial blood, and relaying this information to the center to regulate the cardiovascular and respiratory systems. The carotid sinus, found at the branches of the common carotid artery, is an enlarged area with many morphologically specific sensory nerve endings. It detects the vasodilation resulting from an increase in blood pressure and conveys this information to the nerve center to regulate blood pressure. The vestibular receptors situated in the inner ear, muscles, tendons, and joints receive information about body posture, motion, and balance. Temperature receptors detect not only shifts, but also the rate of changes in temperature. Certain animals are unresponsive to the extent of temperature change due to greater resistance to heat, but are sensitive only to the rate of temperature change in their environment. For instance, in the boiling frog experiment, the frog's temperature receptors establish high thresholds for the rate and degree of temperature change. Consequently, the frog does not promptly escape from gradual temperature increases. The University of California, Santa Barbara researchers discovered that rapid temperature increases of 25 degrees Fahrenheit prompt fruit fly larvae to twist their bodies. The experiment demonstrates that information about the pace of change can be perceived by receptors and transmitted to neurons based on an instinctive threshold response mechanism, which conveys thermal change data to the brain. However, it remains unclear whether the thermoreceptors called TRPA1 proteins directly detect the rate of temperature change, or sensory nerves transmit received information to the central nervous system for processing by specialized receptors, such as injury receptors. Researchers have identified a pore-forming protein known as transient receptor, potential ion channel, TRP channel in living organisms. The ion channel establishes and controls a small voltage difference between the inside and outside of the plasma membrane, by enabling specific ions to pass through the channel. When the body temperature goes beyond 110 degrees Fahrenheit, 43 degrees Celsius, the ion channel opens, activating a sequence of reactions that transmit information via a nerve current to the nerve center that perceives the temperature change. How does this ion channel function? Temperature is the intensity of the thermal motion of an object's molecules, according to the Brownian motion phenomenon. It is essentially the characterization of the degree of motion of another microscopic particle, which will be explained in a later section. Receptors may acquire temperature information through the experience of ion channels that open or close in response to the impact of microscopic particles. Whether the channel opens or closes depends on the strength of impact. The ion channel acts as a single door device, with multiple devices in each receptor and corresponding thresholds for different tapping strengths. If an ion channel, TRP1, responds to the knocking action of microscopic particles only in the temperature range below 17 degrees Celsius, then the change in the temperature interval can be accurately monitored by TRP1 by examining the potential difference after closure. If we consider another ion channel, TRP2, it only responds to temperature changes above 20 degrees Celsius. In case of an on response, it can be detected that the temperature has increased above 20 degrees Celsius. The difference in turn, on response time between two adjoining temperature zones, is crucial to detect the temperature change rate. A potential difference corresponds to each on response, and multiple on responses indicate changes in the potential difference. The ion channel converts variations in the monitored electrical potential over time into temperature data, which is then transmitted by the channel neurons to the central neurons. Subsequently, the low-level nerve center determines the rate of temperature change. Channel neurons act as conduits for direct information transfer between biomolecules and are a vital component of biological and instinctual mechanisms. Other receptors may exhibit similar functionality to this ion channel device. The chain cat shark was chosen as an experimental subject by David Julius and his colleagues at UCSF to study the molecular basis of electroreceptors. The analysis revealed that the electroreceptive cells of the chain cat shark possess voltage-gated potassium channels that are comparable to transient receptor potential ion channels. In response to weak and transient electrical stimuli, these channels generate large, repetitive membrane electrograms. Information response mechanism. Not all information received by receptors needs to be completely converted or transmitted to the sensory system for processing. Organisms have a selective response to information related to energy intake and lock-in strategies. Mechanical, chemical, light, temperature, sound, and other receptor systems convert information into action potentials. The action potentials are transmitted to various regions of the sensory system, including the sensory cortex and nociceptors, through one or more sensory neural pathways. To process complex combinations of information, the sensory system must utilize the brain's multiple processing capabilities to determine the appropriate sequence of responses in a predetermined order. Due to the immense diversity and complexity of the information, how does an organism discern what is advantageous and disadvantageous? What is the extent of the benefits or harms? How do organisms respond based on their evaluation? Each species develops its appropriate and effective mechanisms to interpret and respond to information, including fleeing as the best of the 36 stratagems. These mechanisms are stimulated by the same information for an extended period of time. For instance, by mutating to enhance tolerance to specific information, by learning to synthesize new molecules through trial and error, by adjusting the structure or energy levels of certain energies in the body, by changing habits, moving elsewhere, and more. In summary, diverse information management scripts are offered and performed simultaneously in the species arena. According to Daniel Shamovitz of Tel Aviv University in Israel, in his book, What Plants Know, plants can sense various information from their surroundings using senses such as sight, smell, touch, and hearing. Plant perception research has made considerable progress since the 1970s. Heidi Apple and Rex Cockcroft of the University of Missouri discovered through their experiments that playing the sound of caterpillars gnawing on leaves can cause plants to emit considerable amounts of chemical defenses from their leaves. Despite the absence of eyes, plants such as Arabidopsis have at least 11 photoreceptors. Humans, on the other hand, have much fewer types, with only 4. This implies that the visual system of plants in some ways is more complex than that of humans. Plants possess sensory systems, characterized by various priorities. At the break of dawn, the plant's day commences with the processing of light's information before the occurrence of photosynthesis. As stated by Shamovitz in his book, plants view light as both food and a message. In general, plants perceive variations in temperature by inspecting the magnitude of the temperature alteration in their surrounding environment before responding to it. The appropriate response mechanisms to deal with temperature change-related damage are activated only when a threshold is reached. Plants cannot take shelter like animals in caves when exposed to low-temperature stress. Plants can sense the low-temperature signal in their environment with speed and accuracy to reduce the damage caused by low temperature. As per most of the current research, plants primarily sense the temperature signal changes through their cell membranes. When the external environment's temperature reduces, heat dissipation in plants causes the formation of non-lamellar multilayered membrane structures of lipid molecules on the cell's membrane. This transformation changes the fluidity of the cell membrane from a liquid crystal state to a solidified state. Upon sensing the cold stimulus, the receptor proteins on the membrane change their conformation, activating the cold response signaling pathway, allowing the plant to respond swiftly to the cold stress. Calcium ion channels and two-component phosphate systems are critical in responding to temperature variations. The rapid inward flow of extracellular calcium ions during cold stress increases the calcium ion concentration in the cytoplasm. The plant's response to temperature changes is regulated by changes in calcium ion concentration, acting as secondary signaling molecules. Furthermore, the two-component phosphate system acts as a temperature receptor. The associated kinase receives the low-temperature signal and transmits it downward to stimulate the corresponding resistance. Numerous animals spend their complete lifespan in the open air. How do they protect themselves from the harmful effects of sunlight? According to researchers at Oregon State University, the animal's own ability to make sunscreen may hold the answer. Many animals use a molecular pathway to create compounds that protect against UV radiation, as discovered by the team. Gadusol is the compound that was first discovered in the skin of Gadus morhua and later found in many other fish, amphibians, reptiles, and birds. It provides protection against ultraviolet radiation, which cannot be synthesized by humans and other mammals. The sleeping chironomid is an exceptional species that inhabits semi-dry regions of Africa, and its larvae exhibit substantial resistance to desiccation. Once the larva's body loses 97% water content, it enters dormancy. When the water content exceeds 3%, the larvae resurrect, aiding them in surviving the harsh dry season. In 2002, Japanese researchers discovered a desiccation-resistant PV11 cell line from inactive chironomids. These desiccation-resistant cells can be stored by drying at room temperature and retain the capacity to replicate after treatment. Scientists at Keio University, the Riken Institute, and other establishments found that PV11 cells eliminate the effects of reactive oxygen species when dehydrated and restore damaged DNA caused by desiccation during water restoration. As a result of this drought-resistant technology, the probability of larvae surviving the arid season has been enhanced. The majority of multicellular organisms excrete chemical molecules, which we classify as pheromones and use for messaging purposes. Members of the same species detect specific types of pheromones secreted by other individuals by using their olfactory organs, e.g., olfactory bulbs and plumose nasal apparatus. Behavioral, emotional, and physiological mechanisms can change according to the concentrations of these pheromones. Social insects, such as ants and bees, release alarm pheromones when their nests are invaded to send alarm messages and alert other members to respond immediately with vigilance. During mating season, some species release sex pheromones to attract all suitable mating partners within a large radius. Sex pheromones in non-group living animals have high concentrations that can be carried by the wind over long distances, up to 2 miles. There is also a priming pheromone, but it is less effective between two individuals. The physiological response induced by the pheromone is more delayed, and it does not necessarily lead to behavioral changes. The Lee-Boot effect provides a typical example. In rat colonies without males, females release priming pheromones that consistently trigger responses in their secretory glands when sufficient concentrations are reached. Over time, all female rats' menstrual cycles synchronize. This mechanism may increase the concentration of sex pheromones in female rats during estrus, which could spread these pheromones farther and attract males from greater distances for mating purposes. Mechanical contact stimulus on the body surface creates a sensation through pressure and traction forces acting on tactile receptors. When an external force acts on the skin continuously or with increasing intensity, the sensory system differentiates between two sensations by producing nerve discharges. The nerve discharges in response to continuous stimulus are known as pressure sensations, while the small and non-continuous ones are called tactile sensations. The sensory system responds to different discharge patterns in a particular sequence, with a slow reaction to pressure and a fast reaction to touch. This reaction sequence may be because the nerve fibers that sense touch are thicker in diameter and more sensitive to touch than pressure. Tactile sensation is a fundamental sense that occurs widely in the animal kingdom and can stimulate basic, non-localized motor responses, such as bodily contraction and curling. It can also provoke defensive responses such as whole-body rigidity, body rebound, sluggishness, and avoidance. Microorganisms also respond to information through unique sensory mechanisms. For instance, pathogenic bacteria that infect humans can detect DNA fragments from deceased members of their own species. Overuse of antibiotics considerably alters the concentration of DNA fragments from antibiotic-killed bacteria in the human body. Pathogenic bacteria promptly develop resistance mechanisms by exchanging numerous DNA fragments to isolate potential resistance genes from dead bacteria.